So welcome to the third session of the third annual Women in Science IUFRO webinar series. My name is Josephine Kefelec. I am a forest entomologist specialized in the reproductive ecology of invasive insects. I also served as the deputy coordinator for the entomology research group within IUFRO Division 7 on forest health. Me and the rest of the team are so very excited to have you all here today, and we wish you a happy International Day of Women and Girls in Science. We are continuing our two-day event with yet another fantastic panel. All the women you will hear from today are influential trailblazers in their fields, and we're very excited to hear from them. If you have not heard of IUFRO before, this event is organized by the International Union of Forest Research Organizations. IUFRO is a nonprofit, non-governmental, and global network for forest science cooperation. Since its foundation, IUFRO has been influential in advancing forest research and promoting worldwide collaboration between scientists across all fields of forest research. This event is coordinated by the Division 7 Forest Health in collaboration with ForGEDI, the task force that you will hear about later during this session. If you would like to get involved in IUFRO activities, we invite you to visit iufro.org to find out about events or contact office holders so you can participate. I would like to thank my team for organizing this organizing this event with me. They are an absolutely fantastic group of people, and I feel very privileged to have worked with them for the last few months. During the webinar, please leave your cameras off so to preserve bandwidth. Use the chat for any communications and to ask questions to our speakers. Our moderator will ask them for you. And this meeting is being recorded and will be posting on YouTube after the event. And finally, closed captioning in English is available in this meeting. You may have to activate them by clicking the caption button at the bottom right corner of your screen. And if it is not currently visible, you might have to click on More. In parallel to the webinar, we also created a survey for you to fill in. It is open to all gender, and it is related to the experience of women in the field of forest sciences, as to give you a voice and to understand the barriers that are preventing women from securing jobs in the field. Finally, let me introduce our moderator for this session. Ariska Vendernest is a postdoctoral fellow at the Forestry and Agricultural Biotechnology Institute at the University of Pretoria in South Africa. Her fellowship is part of a collaborative effort between FABI and the Royal Garden Eagle Group based in Indonesia. Take it away, Ariska.
Good morning, afternoon, and evening, depending on where you are in the world. Josephine, thank you very much for the introduction. So welcome to the last session of the third IUFRO webinar celebrating the International Day of Women and Girls in Science. In this session, we have 5 phenomenal women talking about their research in forest health, and this session promises to be very diverse as we will touch on topics in relation to forest pathology and entomology, forest genetics, and sustainability and transformative change in the forest sector. If you have questions throughout the session, please type them in the chat box so we can attend to the questions after each talk. So our first speaker for the session is Dr. Jess Hartshorn, an associate research professor at Central State University in Southwest Ohio. She studies invasive species and disturbance in relation to forest health, and in addition is also a student in mental health counseling and incorporates psychological principles into her work on natural resources. She also helped establish the Queer and Life Sciences Registered Student Organization at Clemson, and she uses her voice to advocate for women and minorities in STEM. Over to you, Jess.
Okay, am I unmuted now?
Yes.
And everybody can see my screen?
It's not in full screen, but
awesome.
we can.
Okay, awesome.
There we go.
Yeah, thank you guys so much for inviting me to talk. I think one of my master's students, Pravina Sharma, presented in this maybe a year or two ago. So I'm really excited to talk to you about some of the work that I did with a group of IUFRO scientists, very similar to the survey that Josephine just shared. So I think it's very well known now that enrollment of women really across the board has increased in college for bachelor's and higher ed degrees over the last 20 plus years. Currently, women actually make up more than 50% of bachelor's and higher ed degrees, and that is true for STEM fields as well. But the issue is that this doesn't translate to professional positions after the end of whatever higher degree they're obtaining. Historically, this has been referred to as the leaky pipeline, but there's a lot of problems with this analogy. The first and probably most obvious is that it's very oversimplified. It emphasizes a single universal experience that is finishing high school, going to college, getting your master's, PhD, and going on to a professor role, possibly with a postdoc in there. And we all know that that's not the only pathway that people can take within STEM. And we know that that's not the only position or job that's available for people with higher degrees in STEM. So this analogy ignores a lot of alternative pathways and professions. It also tends to put the blame on women who are leaving. So it looks at why are women and minorities leaving STEM rather than looking at it as what are the systemic issues and gatekeeping that are pushing people out of STEM. And finally, there's really no room for intersectionality, just like it's overemphasizing this single universal experience. My experience as a white woman moving through STEM is not the same as a Black woman moving through STEM or a Latina woman or an Asian woman or any other woman that comes from a different background. And so a lot of problems with this leaky pipeline analogy. Oh, and now it's not. There we go. Within IUFRO, so you guys will hear about this more later, there is already a task force. IUFRO is actually one of the societies, scientific societies, that's already really active in terms of promoting women in forestry positions. There's the webinar series like this one that we're on right now, participation and sponsorship of international conferences, and so on and so on. Despite this, all of this progress, though, there hasn't been as much progress in agriculture and forestry professions. We know that we still experience a lot of overt and also covert sexism, as well as things like pay inequality and assumptions of incompetence. So, like for the last point, a good example is anytime I go to a conference or a workshop or a training, and I'm with a male colleague, even though we've been in the same field for the same amount of time, we both have tenure, we both have students, all these things. I couldn't tell you the number of times that somebody has assumed that I was somebody else's student or technician. That kind of treatment by people can really wear away at you over time. So this just kind of ties back into that leaky pipeline where we know that it's not like I would never want to leave STEM. I love STEM. That's why I'm in it. I love forestry. But little microaggressions like that over time can really wear a person down and wear down their sense of capability or their self-confidence. So I floated this idea to some other members of IUFRO in the leadership. We developed a team that spans entomology, pathology, both the northern and southern hemispheres. We've got Mariella Marzano, who's an amazing social scientist. So I think we got a really good team together to kind of examine these alternative pathways and challenges in forest sciences. So our objectives were to first kind of identify the major issues that women are facing as professionals in forest health or forest health fields. We also wanted to look at the views and attitudes towards women in professional roles and look at how those attitudes and challenges and opportunities have changed over time. And then finally, kind of the end goal for this was to provide suggestions for organizations to use so that they can better support women in these fields. So we developed a survey that looked at 7 different categories, including challenges that people might face related to their gender or not related to their gender. We also asked about opportunities for improvement, as well as what kind of support and needs people are experiencing. Just as a sidebar, we also sent a survey to forestry departments in the United States and also looked at the leadership, the demographic makeup of leadership in IUFRO. Due to time, I don't have time today to talk about those other 2 components. But if you're interested, the paper's already been published, and you're also welcome to contact me. So we got 129 responses. They were from people across the globe, but most responses were from scientists in North America and Europe, which is probably somewhat expected. We did have most of the respondents were women, but we did have a healthy serving of men in there too, which is awesome. Nobody identified as transgender or said that they did not want to disclose their gender. So we did have that gender makeup for all completed responses. Probably the first thing that jumped out to me was that the women that responded had been in the field for less than 15 years on average, whereas the men that responded had been in the field for more than 20 years on average. So this could relate to that shift that we were talking about earlier, with women making up more of the bachelor's and higher ed degrees and kind of moving into those professions. Women on average tended to belong to fewer societies, whereas men belong to more. We know how expensive it is to just to be a member of these societies, not including actually going to conferences or participating in workshops or some of the other pieces of that. And women tended to do less service for scientific societies, while men did more for societies. However, on the flip side, women did more institutional service. So, like internal, being on committees within their department or their college, versus men who tended to do less institutional service. Anyone who is familiar with at least the United States tenure track system and how that works knows that society service is valued much more than institutional service. Because society service, if you're a chair of a committee, if you are serving on an editorial board, those things you can add a lot of weight to your tenure package or to your CV. Whereas institutional service is really not valued very much outside of your institution, and sometimes not even inside of your institution. And then there's this idea of tokenism that comes along with that too. So when there's fewer women in a department, so like for example, I was the only woman in the forestry program within my department for over a year, between one woman retiring, so we had 2, and then there was just me, and then we hired another woman. So even at the max, there were 2 women across the whole forestry program. And so the road to hell is paved with good intentions. People want committees to have female representation, but in doing that, me being the only woman, then I was being asked to be on every single committee that existed. So this idea of tokenism can really put an undue service burden on women. And this is even worse when we talk about the intersectionality. Black women experience this at an even greater level than white women, of course. Oddly, at least to me, women were less likely to be married and less likely to have children, whereas men were more likely to be married and have children. When you take a step back and you look at the load that women and men have to put into their families respectively, this might make a little more sense that, women who are focusing on their career are delaying marriage and children. However, despite the fact that fewer women were married with children, they still reported less career satisfaction versus men that reported higher levels of career satisfaction. And this was across the board, whether they were single, married without children, or with children, or married with children. And then finally, more than half of the women that responded reported challenges specifically related to their gender. So experiencing firsthand some of that overt and covert sexism that we talked about a couple slides ago. This is in comparison to 2% of the men who responded that said that they had experienced gender-related challenges specifically. Women also reported higher levels of isolation and higher levels of internal conflict. So when I say internal, I mean, again, like within their department, within their college. And men tended to report more external factors as challenges. So just kind of general competition within the field, competition for grants, things like that. Something that was really interesting to me was that women tended to report very specific and very intentional reasons for applying to jobs, whereas only men reported opportunity as a reason that they got a job. So, like, it was open, so I applied for it, was one of the quotes that we pulled from the surveys. That kind of opportunistic entry into the field was never reported from any of the 76 women that responded. And I think really the most important takeaway that I got from this survey was that both men and women report that these shifts in the workplace culture, which allow us to have these open discussions, has positively affected their career and their career satisfaction. So when we have these events, these webinar series, and we might be focusing specifically on women, but the act of kind of putting these issues out there and making it okay to talk about doesn't just positively affect women, it actually positively affects everybody in the workplace because men are also caring for aging parents or dealing with problems at home. And so men are also now feeling like they're more able to talk about these problems, to request support from administration or their colleagues. So even though we're focusing on women today, really the benefits are there for everyone. So some of the suggestions for improvement, really this comes down to that last point I was just making is organizations should just be very open about these issues, encouraging discussion, creating statements that talk about commitment to diversity, equity, and inclusion, because it really just opens the doors for everybody to be more open, and that is going to improve that work-life balance and make people feel more satisfied with their career. It's also really important to promote these leadership positions, especially within scientific societies for women, but also to be cognizant of that extra service burden and that tokenism that I mentioned earlier. And finally, kind of maybe a more minor thing, but something that can have a disproportionate impact, I think, is including childcare and family activities, at least just in consideration for like organizing conferences or meetings. Even something as simple as providing a list of family activities to do outside the conference, even if nobody participates in those, can still make families feel more welcome and also make scientists who are traveling with their families feel like, okay, this is a place for me. I can bring my spouse, I can bring my partner, I can bring my kids, I can bring my family, and I'm still at the table, I'm still being considered. And finally, the one that came up probably most often was when we have these token women who get brought to the table over and over, asking them who isn't here and should be. Because those are the people that are going to be connected to that extended network, just like we're all connecting right now. And so, if we're not asking the women who are already there, like, who's missing out on this? Who should we be inviting? We're going to miss out on a lot of really great talent. So ultimately, valued leadership roles are really necessary and should be a priority for institutions as well as societies. We need a lot more visible support, which IUFRO is doing a wonderful job of already. Consideration of childcare and family care kind of across the board. And then we have to be really intentional with it. So we know just saying something doesn't mean a lot if we don't back it up with the actions. So there has to be this level of intentionality with it. And unfortunately, I have to go teach class. So I can't stick around for questions, but my email's up here. And also Josephine can share my email with you guys too, if you want to reach out or have any questions or comments afterwards.
Thank you so much, Jess. I know you're running to class, so
Yeah, I'm so
Josephine just
sorry. I have to go
Josephine,
teach about life tables, if that makes anybody feel better. We're gonna
um,
struggle through this, so But yeah, thank you
Josephine
guys
just
for having
posted
me.
thank you for coming. Josephine
Appreciate
posted
it.
your email address, and then also if anyone has questions, post it in the box and we can also forward that to you. So thank you
All right.
very much for joining
Thank you
us
guys
today.
so much. Bye.
Bye. All right, so we'll keep this question for Jess for later. And move on to our next speaker, Dr. Violet Butterwort, who is a forest health researcher specializing in insect-vectored forest diseases. She has worked in a variety of roles across her 16 years in entomology research, including academic, agribusiness, museum, nonprofit, and government research positions. In her current role, she performs forest pathology diagnostic services and conducts research on emerging tree diseases in Canada. Dr. Butterwort is an involved community leader, leading walks and workshops on mushroom identification, queer ecology, and wild food. She's deeply passionate about worker and human rights and serves as vice president of her local union. Over to you, Dr. Butterwort.
Thank you so much for that introduction. I'll just pull up my presentation. Okay, is that visible full screen?
It is.
It's on screen, please.
Thank you. All right, thanks everybody for having me here today and listening to my talk. My name is Violet Butterwort, and I'll be talking about some really cool beetle symbioses with forest pathogens, and just kind of a lot of research that I've done over the years. I'll be focusing on some of the research I've done for my master's and PhD, and then a little bit of what I'm doing right now in government research. So some of the beetles I'll be talking about today include ambrosia beetles, the rest of the bark beetles, and sap beetles, and their relationships with forest pathogens. So I'm going to go through these one at a time, and I'll start with the ambrosia beetles and some of the work I did during my PhD on them. So if you're not familiar with this symbiosis, this is what it looks like in nature. It starts with a beetle that bores into typically a dead or dying tree, and it's there that it introduces a fungus that both the adults and the larvae must consume to survive. Once the larvae reach adulthood, they leave the beetle gallery and they bring the fungus with them. And the way they do this is through this like specialized fungus pocket called the mycangium. So if you look at that photo in the middle, you might see like some concentric circles underneath the cuticle of the beetle. So if we do a CT scan of this beetle, it looks like this long tube that winds up underneath the cuticle. And this is where it's storing the fungus. And it's doing this to take it to a new tree and start a new fungus garden. And often you'll see this as these little frass or sawdust noodles being kicked out of a tree. And these insects are one of the most frequently introduced insects around the world. So this figure is depicting US bark and ambrosia beetle introductions over time. And a few of these species cause harm to non-native ecosystems. The vast majority of them are harmless, but a few of them do cause disproportionate harm. So one example of that is the beetle Xyloborus glabratus, and one of its fungal symbionts is Harringtonia loricola. So this has been a devastating disease called laurel wilt in the southeastern United States. And the way it works is the beetle introduces the fungus into the tree, and it kind of causes almost like an allergic reaction where the tree cuts off its vascular system and the tree dies. And this has caused a lot of harm where it's been spreading. It may be resulting in possible tree extinctions like that of red bay and swamp bay. And there's also a lot of risk to other Lauraceae species in Central and South America as it spreads. A lot of these host species also are the host species of butterflies. So it's possible that these other insects will be affected as well. Red bay and swamp bay are also a very important tree to the Miccosukee and Seminole peoples of the southeastern United States. They're used to make handicrafts and in ceremony. And finally, we've seen some loss of yields to the avocado industry as they're also in the family and occasionally attacked and affected by this disease. So one of the questions that I wanted to address was, can this fungus spread to other beetles? But even more broadly than that, how specific is that partnership across ambrosia beetles? And how do they maintain this symbiosis? So to do this, I did a lot of culturing of beetles. I also did high throughput amplicon sequencing of beetles using the MySeq platform from 9 different evolutionary origins of this fungus farming habit. And I did this both for the fungus pockets, the mycangia, where the beetles are storing the fungus. So I would dissect those out and sequence them or culture from them, but also from the beetle galleries. So that way we can connect what the beetle is transporting with what's actually growing inside the wood. And then I used maximum likelihood phylogenetic analyses to identify the fungi, indicator species to figure out what which of those fungi might be more likely to be the persistent symbionts of the beetles, and then a PCOA to correlate some of those beetle and fungus traits to see maybe how this symbiosis is mediated. So here I'm showing some of the community sequence DNA data. So on the left-hand side, we have the beetle taxa. We have 2 taxa shown right now. On the right-hand side, we see the fungal symbiont taxa. So each shaded bar within this represents a different fungus, and the size of the bar is depicting the percent read abundance, or the percent DNA reads attributed to that fungus. So the ones that are grayed out were not found to be a significant associate from the indicator species analysis, and the bars that are colored were found to be significantly associated with the beetles. So now here's more of the data, those data. And some of the takeaways here is that we were able to identify the fungal symbionts for the first time from 16 beetle genera. In general, found that there's typically 1 beetle genus associated to 1 fungal genus. So you can only see like 1 fungal genus depicted on the right in general. And kind of confirming some previous studies, finding that fungus farming evolved at least 15 different times within beetles, and beetle farming evolved at least 12 different times in fungi. A really cool part of this study was also finding lots of new species of fungi. And even when they're in culture, I do think fungi are charismatic, especially the ambrosia fungi have lots of cool different smells and morphologies. So that was really fun. So when we map these phylogenies together, we can also find a few more interesting findings. Like we're kind of confirming that there's these 1-to-1 pairs that seem specific at genus rank. And also just to clarify, the solid lines are depicting data that was obtained through culture and sequence data, whereas the dotted lines are just from community DNA sequence data. But this doesn't quite answer like how specific they are at a species level. So I'm just going to talk about one clade next in a little bit more detail. And so this is another study that I helped with where we looked more specifically at beetles and fungi within this like smaller symbiosis. And there did not seem to be co-cladogenesis that is often found in really, really tight-knit symbioses. So there is some diffuse specificity, but they are still very specific. And it is still, for the most part, a 1-to-1 symbiosis. But a really cool thing I want to highlight is these beetles highlighted in the purple box. So these are kleptoparasites. And if you look at the photo on the left, you can see what I mean. The large beetle comes in first and establishes this fungal garden, and then these smaller beetles come in later and start to burrow in next to the big beetles gallery. And so they parasitize off their fungal galleries. Sometimes they even make their way into the larger gallery and kill the larger beetle. So this is interesting because those are the only cases where we're seeing more than one fungus paired to more than one beetle species. Okay, so how are these relationships maintained? I won't get into all the details here. There's a lot to go through, but I just want to talk about how these fungus pockets or these mycangia are probably mediating this relationship. So there's a few fungi that seem to be really specific, and they're often found in these more complex or larger mycangia. So we fed beetles their native symbiont and then more distantly related symbionts, and we found that over time, the probability of their mycangia filling up with those different fungi is more likely if they have their native symbiont compared to more distantly related symbionts. And I just want to go through some of the cool features of these mycangia because there are so many of them. There are some right behind the mandibles. There are some behind the prothorax. Some of them are these really simple pits on the beetle's back. There's even one Xyloterinus beetle that has two different kind of mycangia, and each one has a different fungal symbiont, which is just so cool to me. I also did some micro CT scanning to help visualize some of these mycangia better. So you can see one example of this. This is Primnobius. And here's a microtome cross-section where we can see those mycangia represented in the same way. And interestingly, a lot of these mycangia have these gland cells, which are probably involved in selecting different fungi. Just more cool examples in different species within this genus. The mycangial tubes have different patterns, sometimes they spiral and sometimes they switch back. And again, these have those specialized gland cells, which you can see as looks like a lattice on the bottom middle photo on the right. And just another cool example, you can find the fungi sometimes just pouring out of these mycangia. So they might be being fed in some way by the beetle. And these ones that are quote unquote more simple or may not have as specialized gland cells seem to be slightly less specific. So most of them are specific in some way or another, and there's multiple factors that modulate the specificity, with mycangia probably being the most important ones. But an important finding is that most of these ambrosia beetles aren't likely to form a new persistent association with plant pathogens. So some of them might pick up that laurel wilt fungus for a period, but over time it doesn't seem to persist in their microbiome. So what about the other bark beetles? These ones are not strict fungus farmers per se. They still have mycangia, but they're often eating some combination of fungus, phloem, and xylem. So one example is the Dutch elm disease fungus. And a question I was interested in during my master's was, are there similar examples of beetle-associated fungi like the Dutch elm disease fungus and the elm beetle, bark beetle, that haven't yet been transported across the globe? So to assess some of these non-native beetles and fungi for risk, myself and others collected a bunch of fungi from bark beetles in Europe and Asia, specifically ones that had some host specificity to pines and oaks. I isolated the fungi from the beetle bodies and galleries, and then I inoculated them in North American pines and oaks inside a quarantine greenhouse. So this is similar to some of those sentinel studies where they're looking at what pests and pathogens are attacking trees when they're planted in non-native regions. But it's kind of like the inverse. Okay, so here I'm showing a lot of those fungal isolates from pines and oaks. And this is, the bars are depicting the percent of stem length that was stained with the fungus after inoculation. So the bottom bars in blue are the negative controls, and the gray bars are a positive control. The ones in green we are calling non-pathogenic because their lesion length was not found to be different than the negative controls. The ones in yellow we're calling mild pathogens, which may warrant further monitoring because they did have a lesion length that was much longer than the negative control. And some of these are also already known to be mild pathogens from previous literature. But thankfully, we didn't find any what we might call a systemic pathogen or a vascular wilt disease pathogen. So if we look at Dutch elm disease or laurel wilt, for example, they will stain almost the entire length of a tree if they're inoculated and under similar circumstances. The other thing I really liked about this study is we could gain a lot more ecological information about these beetles and fungi. So it's really not just a risk analysis where one of my favorite parts was just like learning about the ecology of these beetles because there are just so many of them and they're so diverse. Okay, I'm gonna switch gears a little bit really quick and just talk about sap beetles, which are even more different than the bark and ambrosia beetles. They have much more diverse diets. They're less specific to fungi. And to my knowledge, they don't have any of those mycangia or those fungus pockets. So for example, some of them are fruit eaters, some of them like flowers, and some of them like fungus. These all happen to be things that I love also. But yeah, even the same beetle species will feed on all of these different diets. And some of them can transmit pathogens, like the oak wilt fungus Bracciella fagacearum. So again, really similar to these other bark and ambrosia beetles where occasionally they'll just transmit a vascular wilt disease. So the way this works is we have an oak tree that's killed by oak wilt, and as the fungus grows under the bark, it like cracks the bark and creates what are called pressure pads. So you can see an example of that on the right. And the beetles crawl into those cracks and eat the fungus and sometimes lay their eggs there. And then the way it gets transmitted to a new tree is through a wound on another oak. So they require a fresh oak wound to transmit this disease. So it's like really, really specific circumstances where the beetles can do this. So something I'm working on right now is trying to figure out how these beetles are vectoring this pathogen, which beetles are doing it, when transmission occurs, and what is this high risk period during the year where we can recommend like pruning guidelines so that people aren't pruning oaks during a time when beetles can transmit the fungus to those pruned trees. But I think I'm out of time, and this is something I'm still working on, so you'll have to stay tuned. Sorry for the cliffhanger, but yeah, this is another cool system with beetles and fungi. And I think it just like is a great illustration of the complexity of these systems and the importance of species interactions and community interactions in the context of forest diseases. And I love this complexity, and I think it's such a fun part about research. So thanks to my collaborators, and I don't know if we're taking questions now or later or both, but both are totally fine with me. So thanks for listening.
Thank you so much, Violet. Really, really excellent talk. And thank you for leaving us on a cliffhanger. So we will just have to invite you back to hear about your progress next. So there are a couple of questions in the chat box. So first, Pascal said, excellent talk, Violet. Do the beetles secrete some kind of nutrients in their micandria that could feed specific fungi?
Yeah, so that's a great question. We don't really have good evidence for that yet. And my guess is it might be different for different beetles. Like some might be restricting what fungi can enter, and some might be like providing nutrients to the fungus, but we don't really know yet.
Okay. And then Josephine said, thank you for the incredibly fascinating talk, Violet. You mentioned the smell of the fungi. I'm curious about the response of the beetles to those smells. If the smells attract beetles, how accurate is the response? And do they sometimes attract the wrong beetles?
That is a really good question, and I love the smell aspect and the semiochemical ecology of these beetles. It depends on what beetles you're talking about again. So like the sap beetles are really interesting because the oak wilt fungus has a very fragrant smell that attracts lots of beetles. So that one seems less specific. Sorry, could you repeat the question again?
So it was, if the smells attract beetles, how accurate is the response? And do they sometimes attract the wrong beetles?
Yes. So I don't know what a wrong beetle means, but
Sorry, Violet. I was thinking about like the ambrosia beetles and how specific the relationship is, and do they attract the wrong genus that don't have a relationship?
Yeah. So like that's probably how those kleptoparasites are finding those fungi, because they still only like associate with that one fungal genus. It seems for the most part, the beetles are a little bit more cued into like the stage of tree decay in terms of the smells that they're interested in. Like the ones that are going to more live trees are attracted to much different smells than the ones that are going to like dying trees versus really dead trees. So some of them, like there's a spectrum. So yeah, like we don't have a good sense of which beetles are going where quite yet, but lots of stuff is going on.
Does this relate to, you mentioned an interesting term for me. You said beetle farming. So the fungi are beetle farming. So does the smell relate to that they particularly attracted, or how would you define that term exactly?
Yeah, I don't know if that's like a totally appropriate term. But I mean, it's more like they're relying on the beetle for transport to a new substrate. So yeah, I mean, they're still like growing beetles in a way, and technically in terms of biomass, like the fungus is growing throughout the tree and is like a larger organism than the beetle. But they're not like feeding off the beetle. So it's probably not the right term to use, but I'm just trying to illustrate like how many times that relationship evolved within those separate entities.
Very interesting. There's a comment about the oak wilt by Pascal. He says, in Europe, there is an oak beetle that would be an excellent vector of the oak wilt fungus if it was introduced into Europe. And it could be as effective as the elm beetle for Dutch elm disease. So just a comment on that side, yeah.
Interesting.
Okay, Violet, I think we should move on to the next speaker, but I do want to invite anyone in the audience to still post questions, and we can also discuss a little bit further in our discussion time at the end of the session. Thank you very much. Our next speaker is Dr. Quynh, who is a forest pathologist at the Vietnamese Academy of Forest Sciences. Her work focuses on understanding and managing biosecurity threats and other pathogens in both natural and plantation forests, including acacia, eucalyptus, macadamia, cinnamon, and other native species. Over the past years, she has led and contributed to national and regional projects on integrated pest and disease management, forest landscape restoration, and sustainable forest management, often linking field evidence with policy and community practice. She enjoys working with ethnic minority groups, forest rangers, and local communities through training courses and field activities focused on forest health and biosecurity. We welcome you. Over to you, Dr. Da. Quynh, you can share.
Sorry.
There we go.
Yeah. Sorry.
No problem.
Can you see my screen?
Yes, just go to presentation mode. There we go. Perfect.
Can you see my screen? Okay.
Yes.
Hello everyone again. And so my name is Quynh and researcher of Vietnamese Academy of Forest Science. So it's my honor to be invited to a talk in annual Women in Science IUFRO webinar. So today I just want to present my topic, health and disease in cinnamon in Vietnam and how to manage it. And so first, do you know cinnamon is some of the spicy we regularly use for food and something. And first, in Vietnam, we have around more than 300,000 hectares until now. And the main region is located in the north and the center of Vietnam. And Vietnam, we are located in the tropical and subtropical location. So it creates the perfect condition for the pest and disease spread. And so now the cinnamon plantation in Vietnam, we face some problem like dieback, powder mildew, rust, leaf blight, canker, or witch foot, and some other main pests like bug, leaf eating, bug borer, thrip, or longhorn beetle. So why we do this research? Because this cinnamon is one of the most important species in Vietnam, because Vietnam is one of the largest exporter country for the cinnamon product. So it's very important for my country, especially for the people live in the mountainous areas. So we do some research for the pest and disease, and have the local people recognize the pest disease and damage because and help them to recognize exactly the species and how to manage them. So you can see here in the left right is insect damage. It's called due to the leaf eating. It's called the serious damage in cinnamon plantation in Vietnam. And the right hand side is the damage called to the Phytophthora cinnamomide. It's also one of the big problems in cinnamon in Vietnam. So why we do this research? Because for the farmer, sometimes they confuse between pest and disease. For example, in the nursery, sometimes the symptom between and they cannot recognize because when they saw the symptom, they didn't see the insect because they did appear when they called after they called the dentist. So from here, the farmer think that this due to the disease. And so they maybe use a chemical to treat disease instead of pesticide when the tree was infected by pests. So it's not only negative impact on human health and environment, it also is not effectively for control. So it's still cause economic loss. So, so here you can see this not cause disease. This cause to the mosquito bug. And so also in some case, it's also little quite difficult to determine the pest or disease damage. Like, for example, here you can see the leaf symptom like this one. So sometimes the farmer think that this cause to the leaf disease, but actually it's due to the tree damage. So another symptom like this one is they also from the distance or from the distance you can see and many the cinnamon was infected. Some many cinnamon plant died, and the leaf, the sauce and the sauce dried like this one, and so it's not how it's not the disease. It's how to the beetle like this one. So it's very important to this one is very important to taught the local people to recognize the pest and disease in sun cinnamon in Vietnam. So our research, we found it totally 95 species, which 45 identified and more than 10 unidentified species. It's belong to the 21 families and 6 orders, of which we found that 6 species causing the major damage, and 20 species cause the damage, and 69 cause minor damage. And in here is the 6 species cause a serious problem in the cinnamon plantation in Vietnam. First one is the thrift. As you can see here, with the life cycle of the thrift, is from the age, larva, pupa, and the adult, and they call the they always call the damage for the young leaf, young sauce, and the symptom like this one, and the leaf turn to dry, and the death sauce. So the another main species is piranha, is a leaf eating. This cause also the serious problem. They can eat everything, including the leaf, the bark of the cinnamon tea. So another species with the anther is the bark of the cinnamon. You know that the cinnamon species, the bark is the bark is very important because the farmer get benefit from this. They harvest the bark, and this one is the very important product. But now this species damaged the bark like this one. And another species is leaf eating with the leaf eating, and another one also another species, Busula supensista, also is a leaf eating species. And the last one It's also serious problem. As you can see, they can eat everything. It's not only the cinnamon species. Another plant surrounded here also infected by this species. They can eat everything and eat the whole tree, the whole plant from the bark, the leaf, and everything. So another ones we found this new species in Vietnam. This name is the young source of cinnamon, and it's named Polylufa vietnamnise sp. And another species also called the like lip backworm is called also some the serious problem. And another species like lip miter and lip eating. So we're lip eating. So for the pest, we found more than nearly more than 90 species. So this and we try to take the picture and it's clearly and to have and simply and to have with the symptom and to have the farmer can recognize and can report, and so can get the feedback from the scientists for the how to manage them. And another research about the disease on cinnamon in Vietnam, we found totally more than 50 species belong to 23 family and 18 order, in which 5 species causing the major damage, including Phytophthora cinnamomai, Collectotrichum, Podophorus, and Diboter, and Artisidium, and 25 species causing the damage, and 29 species cause minor damage. And here's the picture of pathogen and the symptom they call in the cinnamon plantation. It's a Phytophthora cinnamomai and Collectotrichum greosporidate, and the powdered mildews like this one. And also Diaboter is caused the mist in the nursery, and the rust is called both image in both nursery and plantation. And so how's we prevention and monitor them? Because this one is spicy, we use for food. So we the priority, the methods focused in silviculture, physical biocontrol, and the last one use a chemical. And so for example, for the so for the pet, damage bugs like this one, we normally use swab to remove the insect from the bark. So you can see the swab here. We use this one to remove. And so for this species, for the leaf eating, we use we collect the pupil from, because it depend on the behavior. We collect the pupil from the soil and destroy them. And for the adult, we use a simple technique like the light traps. We use like this one with the water and oils and the light. In one night, we can collect more than 2,000 adult. So it's a simple, it's a simple chap, but it effectively for the control. So for the thread, we use color chap, color sticky chap like this one in the nursery. And so we also conduct the endophyte experiment for the cinnamon, for the control cinnamon disease. And you can see here, there are some experiment we did try to use endophyte from the cinnamon. We found the healthy one and collect and isolate some the endophyte and to test with some disease. And also use nature enemy like this one, Bavaria bassiana, to control some the pests or metasium, or use or maybe spider can use. And so for our research, we found more than 95 pests and 56 disease of cinnamon, and identify 64 pests and 50 species of pathogenic fungi. In our research, we found 37 new record for cinnamon pathogen in Vietnam, and especially we found 2 new species. It's a Phytophthora vanillensis and Phytophthora mandibulatas. It's a damage in cinnamon plantation in Vietnam. And also develop the control technique for the farming community, and progress towards organic production of cinnamon through use management and biocontrol methods, and production handbook in Vietnam and English, and helps the farmer to identify pest and disease in their plantation and the pest control measurement. And so this one is some of the research we did, and we did for our research of pest and disease in cinnamon in Vietnam. So thank you for listening, and any question? Yeah, welcome. Yeah.
Thank you very much, Quynh. It's very interesting to see that there are so many pests and pathogens associated with cinnamon. It's very, very nice to see your work. There's a question here from Violet. She says, very cool study. So how many pests and pathogens? So she said, sorry, so many pests and pathogens, same as I commented, sorry. So she asked, did you inoculate any of the endophytes into trees to assess their effect in a field setting?
Thank you for your question. Yeah, actually, I did inoculate I did the in vitro in the lab, and we just beginning conduct the trial in nursery, but then working for the results, because for the endophyte, it's a slow progress to see something happen. So for the period, for my period research, I found just the root is different, but we still continue to continue to working with this to find how to endophyte work effectively or not with the disease in a reality. Yeah, because it's the slow progress. So yeah.
Josephine wrote, "Thank you for the informative talk, Quynh. I see you use blue and yellow traps for thrips. Do you notice a difference in the efficiency between the two colors?"
Well, for this case, the tips is the yellow is more effectively than the blue one. Yeah.
Why do you then also include the blue? Or does it catch something else or different species?
No, it's then catch something like fly or some other species.
I see.
But the yellow one is more effectively with the trip in
Okay.
nursery. Yeah. And also I saw here is another question from Angela.
That's right. So she's asking, is there any combination of management approaches that will target both the fungi and the pests?
Actually, now to be honest, in Vietnam now the pest is caused a more serious problem than disease, and now we just depend on some case or some real situation. We will apply the exactly the methods. For example, if we found if we taught the farmer how when they saw especially in the summer comes it's moisture and humidity and raining, so it creates perfect for the trap damage. So when they saw the lip starts curly, we start use the treatment. So when you see we use a treatment as soon as possible, it's more effectively. So that's depend on some season, some situation, or some plant is a nursery or young plantation or something. Yeah, something like this one. In online, yeah.
You mentioned at the end that you are publishing a handbook to identify the pests and pathogens. How, what is your communication like with the sector itself? Do you have a reporting system so people can reach out to you, or do you just reach out to everybody on your own, or is there a process in place for people to come to you and report if they see anything?
Oh, actually, we not only we have we have also have the social connection, like we use a Zalo or something or Messenger. We have a group chat. So when the farmer sees something, they can take picture and send the group, and we can help them for identify or something. Yeah. Now for this book, actually, for this book, I got the sponsor from the Asian Center for International Agriculture Research from Australia. They sponsor for us to for this book. Yeah. So it helps the people can use and can easy to recognize pet and disease. Yeah.
Fantastic. Thank you very much for your time, Quynh. Please stay so we can have a discussion afterwards as well. We are going to move on now to Dr. Katharina Budde, who is a biologist specialized in population genetics and genomics of forestry species. She has worked in several forest research institutes and universities in Spain, France, Denmark, and Germany. And currently, she is acting head of the section Forest Genetic Analysis at the Northwest German Forest Research Institute, where she aims to understand evolutionary processes in forestry species in order to develop sustainable conservation and management strategies. Over to you, Katharina.
Thank you very much for the kind introduction. Can you hear me?
I can hear you clearly, yes.
Okay, I will share my screen.
Okay.
Can you see it full screen?
We are still, yes, that's correct. You can
Okay,
continue.
perfect. Then I will start. Okay. Yes, hello everybody. Thank you for having me here. I'm very happy to present a little example of our research here today. So I choose a quite broad title, but actually I brought an example from ash dieback research that we are doing. And most of the work, I started the work when I was at the University of Copenhagen as a postdoc. I had a long time as a postdoc in different labs. And finally, 2 years ago, I got a permanent position here at the Northwest German Forest Research Institute. I know it's a terribly long name, but yeah. So I will present a little bit of what we are currently doing. Okay, it's not moving forward. Just a second. Now, okay, just so that you get an idea where we are, I'm actually pretty much in the center of Germany, more or less close to Göttingen. And this is where our research institute is located. This, what you see on the picture, is our offices in our department, close to the Weser River in Hanmünden. And you can also see the nursery facilities that we have. So at the moment, there is a lot of concern about our forest ecosystems in the changing conditions, right? We can see when we, at least here in Germany, when we drive through the landscape, we can see a lot of damaged trees. There are a lot of damaged spruce trees because of the recent droughts, but also because of mismanagement, because they have been planted in places where they are not locally adapted to. There are other cases, for example, ash dieback. You might know the symptoms. You can see them here in the middle. A lot of ash trees that have quite light crowns because a lot of the branches are dying off. So we have wilting and dieback from the tips of the shoots. And occasionally the trees also die. We also have more forest fires, which was not usually a common thing in Germany, and now there have been reports in the last years that also forest fires become more and more frequent, and our species are usually not adapted to this. So there is a big concern, and there is the question, are our forestry species actually able to cope with these changing conditions, or are they able to adapt? And I just wanted to emphasize that trees have life history traits that might seem not advantages for fast evolution. For example, they are of course sessile, they have very long generation times, but they have other life history traits. For example, usually forestry populations, the natural stands, they are usually characterized by a high standing genetic variation. They also have typically they have far-ranging gene flow by pollen and seed dispersal, especially by pollen. They also, when you think of an oak or a beech, and you think about all the seeds that they produce throughout their lifetime, you can imagine that this is a huge lifetime reproductive output. And you can imagine that, well, to maintain a population, it's sufficient if every individual has one offspring, right? So you can imagine that selection is really strong in early life stages in trees, and this can make allele frequency changes very quickly. So there is a potential to adapt fast, but it is very difficult to detect. So this is a challenge for us because, trees are very long-lived, and to detect the changes when they happen, this is challenging. So I brought one example, ash dieback in Europe. This is caused by an invasive ascomycete called Typhus fraxineus. It's a pathogen, well, actually in Asia where it originates from, it's not a pathogen, it's an endophyte that grows on Fraxinus manchurica. but it has been introduced to Europe in Poland or Lithuania in Eastern Europe, and from there it spread. So it started around in the 1990s, and from there it slowly spread all over Europe. And so it has even reached the northern part of Spain now. And you can see that from the fruiting bodies that you can see here on the rachis of an ash leaf from last year, they form these fruiting bodies and that they release the ascospores. And so when we have a look at the life cycle, so we have first these fruiting bodies on the that release the ascospores. The ascospores infect the leaves of the ash trees, and the fungus grows through the leaf rachis, through the petiole into the stem, and it can cause necrosis and wilting and dieback from the tips of the crown. Occasionally, you also have necrosis on the stem, but this can also be caused by secondary pathogens or secondary infections by other fungi. So, and then the leaves are shed in the autumn, and the fungus overwinters in the leaf rachis in the litter, and next year the cycle goes on again. So what we already know from previous studies is that ash dieback susceptibility, although these two species, the pathogen and ash, never co-evolved, there is a genetic component to the susceptibility in the trees. So what you can see here is the damage score, like the health of the crown, in the trees planted into the nest. This is one location in Denmark, and in Tapsør, that's another location in Denmark. So they have planted clonal common gardens, so the exact same genotypes in two locations, and you can see that the genotypes that are doing good in one location, they are also doing good in the other. This is why we have such a strong correlation. So it's really the genotype of the trees that determines how susceptible they are, at least to a big part. So this means that it's a heritable trait. And what we also know from previous studies, when they did meta-analysis of a lot of stands that have been assessed, it was young trees, plantations, natural populations, and they saw that there is a really high mortality in all age classes, especially in young trees. So it's estimated that actually 82% of young trees die. So there is a very, very strong selection pressure on the trees. And we know that trees, adult trees that are less susceptible are more likely to have offspring in the next generation. So they tested this in a population where they looked at the offspring and they looked at the adult trees and checked how healthy were the actual mothers and fathers that were standing there. And they could see that less susceptible ones were more likely to have offspring in the next generation. So this is a good indication why we think that evolution should be going on here. And we wanted to test if this is the case. So we wanted to see what is the effect that ash dieback has in natural populations of ash in Europe? And we wanted to see if we can detect an evolutionary change due to the disease. For this, we had, you can see it here on the map, we had clonal common gardens. These are the how do you call it, the gray symbols. And we had natural populations. These are the round circles. So we have some populations, these are the yellow ones, and these are the ones that we will focus on today, where we have samples from the year 2000 and we have samples from the year 2019. So in the year 2000, they sampled adult trees, and this was before the disease outbreak. So you can imagine that this is just a random sample from before the disease hit. And then we have young trees from 2019, which is a random sample of the young trees that emerged in the presence of the disease. So we used low coverage whole genome sequencing And we had 486 trees in the common garden, 234 trees from the natural populations. And then we checked with our common garden data, where we also had the health assessment of the crowns, like how healthy did the trees look. We did an association study to see which loci are associated to susceptibility of ash dieback. You can see it here. This is a Manhattan plot where we have plotted the p-values. They are plotted on the y-axis, and we have plotted every single locus in the genome. So you see the genome, it's ordered by chromosomes, and you can see that there are a few peaks. So there are a few locations where we have loci that seem to be associated to crown damage, but none of them are highly significant, because none of them surpass the red line. We have significant loci, but not highly significant. So it is a very polygenic trait. So we do not have a single locus that is responsible for tolerance to the disease. So here we have many loci with rather small effects. And we use these loci to build a prediction model. So we use these loci, the best hits in our genome-wide association study, to predict the health score in trees that we do not know yet. So we calibrated our model based on the trees from the clonal common gardens, and then we applied it to the trees that we have from the natural populations. And we predicted the health based on the genotypes in the old trees from the year 2000 and in the young trees from the year 2019. And we could see that in 4 locations, the change is already visible. So the young trees are doing better than the old trees. So there seems to be already a change in the phenotype. And we can see that this is mostly the case in Eastern Europe, which is where the disease started. So most likely there, selection has been going on for longer already, and this is why we can detect it there. So this is a good sign, because there are a few locations where the evolutionary change is already happening. The trees are slowly adapting, but is it fast enough? That we cannot say. But in order to give the species a chance to survive, it is really important to foster natural regeneration, because selection is always most efficient when it's acting on high numbers of individuals. So I think a good message is to foster natural regeneration of ash. We were also wondering, what is the impact of the disease? Because we know that the mortality is so high in these populations, it should have an impact on the genetic diversity of the populations. So here I plotted on the y-axis the nucleotide diversity estimated on the whole genome and the locations. And we have the young and the old trees. And you can see that in almost all locations, we do see a strong drop in genetic diversity from the old generation to the young ones. So there is loss of genetic diversity, except in one location, and this is here in Kaiserdoris. And at the beginning, we were wondering, why is that? Why this population doesn't show it? And it is a location where we have a huge floodplain with many ash trees. So it's probably the size that is buffering. So here, a big ash population is probably not showing this drop in genetic diversity, because there are so many individuals that the population manages to maintain the levels of genetic diversity. But something that we have to keep in mind when we detect changes in genetic diversity in tree species is that tree species, because of their life history traits, it is more difficult to detect this change. So we are probably underestimating the loss of genetic diversity still, because ideally we should compare the adult generation, the next adult generation. You know, we cannot compare the old adult generation with the young trees now. It's not the same. So we are a little bit comparing apples and pears, and we are probably underestimating still the effect of genetic diversity loss. So ideally, we should do it when the trees, once they are grown up. But of course, waiting for this is too long. So at the moment at our institute, we have a new ash project. It's called FoRISK Recovery. So it's already the second one. And in our research, it's a very applied research. So in the first round of projects, they actually identified healthy trees in the landscape and in highly damaged stands. So they assessed the trees over several years, and they took them in, they grafted them, they replicated them, and then in a replicated experimental design, they actually tested for susceptibility using an ascospore inoculation experiment. And at the end, they came up with 96 genotypes that really were less susceptible to the disease. And these have been grafted and planted in a seed orchard. But this seed orchard is now 3 years old. So you can imagine that it's not carrying many seeds yet. So this will take time. But it could be used, for example, to validate our prediction model that we have been using before. So we are currently sequencing the trees to use them also for the genomic prediction. So here is a progeny trial of seedlings from these plus trees. And we also have started to take these genotypes into in vitro culture in order to maintain the genotypes and to be able to produce experimental plants for experiments. And with this, I'm already at the end. And there have been a lot of people involved in this research who I would like to thank, and I would like to thank you for your attention.
Thank you very much for a very interesting talk, Katharina. Question here from Pascal. He says, nice talk. I agree with you that we should foster natural regeneration of ashes in Europe. What is your opinion about breeding ADB-resistant ashes as advocated by some labs?
Yeah. So I think because it's such a polygenic trait, I think there are many solutions to the problem, because there is not a single locus that has a strong effect. So there is not a single locus that we can promote and disperse in our ash populations that will make a change. So it is really a very polygenic trait, and I think we rely a lot on the diversity that is already there. And I'm not so sure. I mean, of course, we can try to select trees also using genetic markers for a breeding program, yes. But I think there is a lot of genetic variation there that we should foster.
Thank you very much for your time. We have to move on to the next talk, but please stay online so that we can continue the discussion afterwards. Thank you very much. Our last speaker, but certainly not the least, is Juliet Achieng, who is a PhD candidate at Wageningen University in the Netherlands, as well as a researcher at the European Forest Institute in Germany. She has a keen interest in global forest research topics, specifically forest-related employment, including green forest jobs, forest education, gender, as well as agroforestry and rural development. She has over 10 years' experience working in international processes and organizations on different continents. She's also the deputy stream leader of Stream 2 of the IUFRO Task Force on Gender Equity, Diversity and Inclusion in Forest-related sectors. And I believe we will hear a little bit about this today. Over to you, Juliet.
Okay, yeah, so thank you so much for inviting me to this webinar. My presentation will be slightly different from what the other speakers have been sharing. So I will highlight two initiatives that I believe are crucial to driving sustainability and transformative change in the forest sector. So these two initiatives are the FoRISK, which is the Forest Risk Facility, and the ForGEDI MOOC course. So I will not share my PhD work today, so I will just focus on these two initiatives. Okay, so I hope you see my screen, but now it's not moving again, so just give me a minute. I stop sharing and then I reshare again. I'm sharing again. I hope you can see it in presentation mode.
We go, yes.
Okay, good. Yeah, so my presentation is focused on these two initiatives that I've mentioned about, but I will start with an overview of the challenges that we are facing in the forest sector, and then I will talk about the activities of the FoRISK facility. I will share how we achieve our goals and how we carry out our activities and who helps us implement these activities. And then I will talk about the rapid response mechanism and why it's important. And then for the second part of the presentation, the ForGEDI MOOC, I will talk about how the idea started until it was successful. And then I will also share the importance of the course and also give you information on how you can participate in the course and why you should participate in the course. So we know that forests are very crucial to achieving economic, social, and environmental goals. However, there are many challenges that forests are facing, like the other speakers have already shared with us, and also the forest sector. So climate change is causing a lot of natural hazards. So it has increased the severity as well as the frequency of natural disasters. So we have seen that there's an increasing frequency of wildfires, there's an increasing frequency of pests and diseases. We also know that these pests and diseases, they cause significant damage to forest health and productivity, so they affect the quality of the products that we obtain from the forests. Then there's also deforestation and land conversion, mostly for agriculture purposes or urbanization, because we see that the world population is growing, or also land conversion for cattle farming. And all these are major threats to forests. And then we also see that there are economic and social pressures on forests. So, for example, there's fragmented ownership. And then there's also the challenge of degradation and biodiversity loss. So we know that overexploitation as well as habitat degradation damage the structural complexity of forests. There's also the changing societal demands on forests. So we know that the society currently is very interested in ecosystem services obtained from forests, but at the same time, we also need forest products. So we see that there's a clash in objectives or demands that are placed on forests. So what should we focus on? What should we prioritize? And lastly, we have equity and inclusion challenges. So we see that a lot of people depend directly on forests, but when it comes to power and decision making, we know that not everyone has an equal say. So there's inequality there. And what I would also like to say is that these challenges vary across regions. So what is a problem in one part of the world might not be a problem in the other part of the world. But we need innovative approaches and solutions to all these problems that we've talked about. And how do we do this? So one of the initiatives that is out there is the Forest Risk Facility. So what is this Forest Risk Facility? So Forest Risk Facility is an implementation mechanism of the Forest Europe high-level political process. It was established in October 2024 after the signatories of this high-level political process endorsed the Bonn ministerial decision. And it started operating in January last year. So this facility is more than 1 year old. And it's hosted at the European Forest Institute here in Bonn, Germany. So why was this facility established? It was established because the signatories in the pan-European region realized that there is need for dialogue about forest disturbances, as well as the need to promote cooperation across pan-European region and the forest sector. And there was also need to do this in line with evidence-based information. So that's why FoRISK was established. So what does FoRISK do? So FoRISK is a key platform that brings together knowledge and practice exchanges across pan-Europe, as well as a platform where you can share science-based information and innovative approaches rather. It's also a main partner that fosters cooperation on risks based on evidence, and it also it's a network that connects national and international actors. So in line with our aims, our activities include strengthening the science policy practice interface. We also promote transboundary cooperation while at the same time utilizing or applying local solutions to local problems. We also facilitate expert exchange. We also provide capacity building opportunities, and we also enhance communication and knowledge sharing among countries and among experts and all the relevant stakeholders. So FoRISK also has 3 expert groups on wildfires, windstorms, and biotic agents. So who can join the expert groups? So it's experts in fire, windstorms, and biotic agents. So the 3 focal areas that have been already shown up there. And the experts need to be nominated by the national focal points and observer focal points. So who are these people? These are signatories and observers of the Forest Europe process. So after my presentation, I will share the links to everything that I'm talking about, Forest Europe, FoRISK, everything that I will share in my presentation so that you can have a better understanding, because I'm sure these processes are not familiar for everyone. And we have the rapid response. So this is a unique tool of the Forest Risk Facility. And why is it unique? Because it promotes active response and at the same time support countries that are facing natural forest disturbances. So this is based on the expertise from the FoRISK network. So the FoRISK network comprises of the Forest Secretariat as well as the expert groups. It also aids countries in developing ad hoc strategies that are tailored towards addressing unprecedented and or acute disturbance events. So I think even the other speakers have already talked about this. So we've realized that the intensity and frequency of natural forest disturbances is increasing. So it's always good to prepare, but there's sometimes we can be caught off guard. So for example, when a windstorm happens, you can never prepare for it. It just catches you off guard. So this rapid response mechanism now comes in handy. We work with countries that have been affected and we help them to now develop strategies to help them deal with these challenges. We also the rapid response tool also provides tailored management recommendations on how to deal with the disturbances as well as emerging issues of political relevance. We also have the FoRISK Intelligence Hub. So this is a repository comprising of curated resources that focus on the 3 focal areas that are relevant to the FoRISK facility. It's beneficial for forest managers, policymakers, researchers, or students. And it enables all the target group to gain practical insights on how to prepare or to respond to disturbances, and also just gives a better understanding of forest disturbances and risks. So here is where I need your help. So we've had a lot of good presentations earlier. So I would encourage the speakers and everybody else who's working in these focal areas to share their resources with us so that we can upload it to the Forest Intelligence Hub so that it can benefit many other people, so that they can also learn from your work. So even materials from the Global South are welcome, so feel free to share them with us. And we are also very interested in practice policy-oriented materials as well. So you can find FoRISK online. We have our website on forisk.org. So I would kindly encourage you to visit our website to learn more about our work. I've really summarized it because of time constraints. And then you can also follow us on LinkedIn. You can scan the QR code or just go to LinkedIn and type our full name. Or you can send us an email at secretariat@forisk.org. So that's one initiative that I think is very relevant. So now I move to the second part of my presentation, and this is the ForGEDI MOOC. So what is the ForGEDI MOOC? So the ForGEDI MOOC is, I think, the first ever MOOC to be developed, focusing on JEDI issues in forest-related sectors. So the aim of the MOOC is to help advance the JEDI principles in forest-related sectors, and especially in education. So to address the gap of how these issues are covered by the curriculum. And why is it important? So we know that there's inequality when it comes to access to education. And in the IFSA open letter in 2021, there was a call for the topic to be addressed sufficiently in university education. And it also helps contribute to the SDG goals 4, 5, and 10. So how did this start? So in 2022, it came up as an idea following the things that I've previously highlighted. And so the idea continued developing. And in 2023, the structure of the MOOC was developed. And then in 2024, there was the production of the materials, and then there was the platform where the MOOC would be hosted. And on 8 April 2024, the course was officially launched on the FutureLearn platform. So in April this year, the course will be 2 years old. So who did all this work? There are 42 contributors who supported this work, and they're from 29 organizations and from 5 geo-regions. So we are really, really grateful to all of them for all the work they put in to make this idea be a reality. So you can see from the logos that there are many partners involved here, and that there was a lot of mobilization that was done. So the partners did not work alone, so they worked in association with University of Padua. There's also SLU, there's the University de Montaut, and IUFRO as well. So the MOOC is provided by University of Padua, and it's co-funded by SLU and FSC. It was launched in April, like I've said, on the futurelearn.com platform. And who is it for? I know you're asking. So this course is for anybody who's interested in JEDI issues. It could be students in forestry and related programs. So you see, it's also beneficial to students in international relations, for example. So programs that are outside forestry. So we have also business and trade, and also bioeconomy. It's also relevant for educators from universities and technical colleges. And it's also relevant for professionals, so anyone from business or civil society organizations or government and public agencies. So in short, I can say it's relevant for everyone who's curious, who's interested, who's passionate about JEDI, promoting JEDI principles. So the course is delivered via the FutureLearn platform, and it's open access and available worldwide. So it's delivered in 3 weeks. As you can see here in my presentation. So the first week focuses on basic concepts of JEDI, and in week 2, the students or the participants get to learn about worldwide case studies, while week 3 offers the participants an opportunity to learn about practical tools for dealing with JEDI issues in forestry-related sectors. So how do you connect with us? So you can check our website, or so these are the details. So I will also share in the group. So you can visit our website. You can follow us on LinkedIn. You can also follow us on YouTube, and you can also send us an email. So I would also like to highlight that the JEDI is now a task force within IUFRO, so you can also be a member of the task force. Just send us an email and you will be included in our mailing list, and you can get to participate and share your ideas on how we can advance and improve this MOOC. So in conclusion, I would like to say, so what lessons do we learn from these 2 initiatives? So to drive sustainability and transformative change in the forest sector, collaboration across regions and actors is crucial. So there's also need to raise awareness based on evidence-based information. We also need political will to support our work. We also need to enhance interdisciplinary skills and capacities. We also need to develop initiatives that create solutions and also keep the momentum going. So one of the speakers talked about intentionality. So I think this is also needed here. We really need to be intentional. We really need to be innovative. We really need to be thinking of solutions. And lastly, it's also necessary to build trust among stakeholders, the different stakeholders we're working with, so that it's easier to come together and come up with solutions. So thank you so much for your time, and I'm looking forward to the discussions.
Thank you very much, Juliet. That was so informative. Are there any questions from anybody in the audience? I will maybe just ask, with the FoRISK program, you invited us to contribute our expertise. So in what capacity, what would you like us to bring to the table?
Oh, okay, so I'm unmuted. Yeah, so like I said, so we have experts who form part of the expert groups. The 3 expert groups are on windstorms, wildfire, and biotic agents. So your countries, if you're within Pan-Europe, your countries can nominate you to these expert groups. But if you're not within Pan-Europe, it's also possible to collaborate with FoRISK, because these challenges we are dealing with, they are not confined to boundaries. They go beyond boundaries. So you can support us by sharing your work with us, and then we can upload it to the FoRISK Intelligence Hub. And then this will also help other researchers, other decision makers, and other people who are interested in the topic to learn about what you're doing in your countries as well. And you can also learn from them, because they will also have their materials in the FoRISK Intelligence Hub.
Excellent. There's a question from Angela. She says, thank you for the talk, Juliet. Is there a success story of an ad hoc strategy for pests and diseases from FoRISK?
So thank you, Angela. like I said, FoRISK is 1 year old only. So yeah, we just started our operations last year, but we don't have an ad hoc strategy for pests and diseases yet. We are working on that, but we have an ad hoc strategy for windstorms. So there was a windstorm last year in Ireland, and the experts and the policymakers from Ireland contacted FoRISK to support with the development of the strategy. So there's such a strategy, but for windstorms and specifically for Ireland, because that's where the problem was. And you can find it on the FoRISK website.
Okay. Another question here. Thank you, Juliet. This is very inspiring. Can you give us more information on the type of case studies that are included in the MOOC or the MOOC?
Okay, so this part of the MOOC was handled by a different colleague, not myself. So what I can say is that please just register for the course, and then you will be able to see everything that is there. So before you start taking the course, there is an overview of all the topics that are covered. So this will be helpful.
Is there a cost involved in attending the courses?
No, it's free. It's free for now. At least the sponsors we have were able to at least cover it for 2 more years. So it's available freely for the next 2 years. But we keep searching for sponsors for people to sponsor it, because we believe that it will have more impact if it's freely available. So we want it to reach as many people as possible. We don't want to limit it. So that's why we're always actively searching for partners to support it. So if you feel that this course is relevant and you feel like you can support, you're also welcome to do so. Yeah.
Fantastic. Thank you very much for presenting this very interesting work to us. We now have a short time period for open discussion. So if anyone still has some questions they would like to ask our speakers of the session, you are welcome to post it. But then also right at the beginning, someone asked, where is it now, to Jess, and perhaps some of the speakers would be willing to weigh in here and give their opinion. So Pragati asked, when one faces situations where their expertise is questioned despite equal qualifications, how do they handle it while maintaining professionalism? Do any of you perhaps have an opinion on this that you would like to share? No opinions. Oh, there is a hand, but okay, so Dodora, could you perhaps post or type what you would like to say as you are muted? Then there's another question here to Violet. The fungi associated with ambrosia beetles must have great competitive ability to guarantee that other fungi do not grow within the gallery. Do you know what traits allow them to be so competitive? Violet, you are muted.
Thank you. Sorry about that. Can you hear me now?
Yes.
Okay. Great question, Josephine. Thank you. So I think there's a few different answers from the evidence we have available right now. One is that they have different metabolic capacities that are probably co-adapted to different substrates. So I didn't go into this too much, but different beetles will go into trees at different stages of decay, and their fungal partners seem to be specialized to those different substrates. So that's one answer. Another is that the beetles themselves do exhibit some like gardening behaviors. So they will remove certain fungi from the gallery that they don't want. They will like clean the larvae if there is any like possible entomopathogens or anything like that. And yeah, I think that's what I can answer off the top of my head right now.
Thanks, Violet. And then to Quynh, most of your pictures depict very dense plantations. Are they monospecific plantations or managed natural forests? And does tree density affect the level of damage from pests and pathogens?
Yeah, actually, it's the cinnamon. This plantation is not the nature forest, and you're right. You can see the young cinnamon plantation in here in my country. They plant the density maybe around, I think, nearly 10. From the beginning, they plant around 10,000 land in hectare per hectare. So it's quite dense. Yeah. So so after 5 years, they cut out some tree, and then because it's for the cinnamon species, they can harvest both. It's not only tree. They can harvest it's a leaf, everything they can use. So you're right. The density is affected when the pest or disease damage is caused a serious problem and spread very fastly. Yeah. That's also the problem. There's also the reasons. Yeah. Yeah. So we try also to avoid them, maybe mix with some another species to plant with another species yeah, because the monoculture maybe sometime, when they affect some pattern disease, they can spread very fastly and cause a serious problem and huge damage. Yeah.
Absolutely. Thank you very much for that answer, Quynh. We have a response here from Tedora, talking from experience on the question that Pragati asked. So Pragati said, when one faces situations where the expertise is questioned despite equal qualifications, how do they handle it while maintaining professionalism? And Tedora spoke out of own experience and said, it's a situation many of us found ourselves in. I can only share my approach to maintain professional tone and support qualifications with achievements and experiences. But many times it does not help. But we can just continue to try our best to continue in this professional tone. So thank you for that response. Katharina, before I ask you a question, I just want to mention that Juliet is posting a lot of case studies now, as well as links in the chat. So please go and have a look at this. There's the crisis management communication collaboration, and then also case studies from the MOOC that are very interesting. So please go and have a look at that. Katharina, what I wanted to ask you, ash dieback is such a severe problem, and the situation seems so dire. If you would find a particular ash that's resistant, how long do you think would it take to actually to restore the landscape to what it is now? And do you think it will ever actually be the same?
That's a good question. So I think, actually there are ash trees that seem to be able to survive. It's a very low percentage. I think the estimates are around 5 to 8%, but they seem to be present in many, many locations. So I think there is hope that there is natural variation present to contribute to the next generation. But also coming back to Pascal's comment from before, I think we have to really help. So by fostering natural regeneration, I also think that breeding approaches are good. I think introducing individuals that are less susceptible for restoration purposes would be great, because in many places at the moment the species is substituted by other species because foresters don't want to take the risk. And if we remove it from the landscape, we do not give the species a chance. So yeah, I think whatever we can do to help to have high numbers of individuals that are less susceptible, but also to foster the diversity that is there, I think this would be great. But I cannot say how long it will take or if it will really be successful, especially knowing that the emerald ash borer is already on its way from Russia moving westwards. It is a very difficult situation. We don't know.
But we can continue trying.
Yeah,
I guess.
yeah, we should.
Absolutely. Yes, I agree. Are there any last comments from the audience or from the speakers, perhaps, before we close the session? Okay. In that case, as we close today's last session, I want to reflect on what women and girls in forestry science truly represents. It's not just about representation. It's also about leadership, innovation, resilience, and transformation. We know that talent is universal, but opportunity is not always evenly distributed. So we, as scientists, mentors, institutions, and leaders, have a responsibility to ensure that curiosity in young girls become confidence, that confidence becomes expertise, and that expertise then becomes influence. So today I want to thank our speakers for sharing their expertise with us and also for staying in time. We are actually on time, so thank you very much for that. And then also thank you to everyone that attended the webinar and for those who continue to support and advocate for women and girls in forestry science. And the future of forestry depends on all of us, and it will be stronger because of women and girls who are helping to shape it. So let's continue this work together. Thank you very much.
Thank you.
Thank you so
Thank
much.
you.
Thank you so much, everyone. It was lovely having you all here.
Bye-bye.